Hi, everybody. It is 2 minutes past the hours, 6 p.m. In Paris, noon in the East Coast, 11 a.m. actually in Rochester, and 5 p.m. in London. I'm Mohamad Mohty from the Sorbonne University and Saint Antoine Hospital in Paris, in France, and it is my great pleasure to welcome you all to this journal club brought to you by the International Academy for Clinical Hematology around the globe and across the hematology field. And for today, the steering committee of the ISCH has selected a paper which was published recently, actually, in the Lancet Oncology by Dr. Uwe Platzbecker from Germany. And the senior author is our guest panelist today, Professor Amer Zeidan from Yale University. And this is about the Imetelstat, I hope I'm pronouncing correctly, in patients with lower risk MDS who have relapsed or are refractory to erythropoiesis stimulating agent. So the IMerge trial, which is a multinational randomized double-blind placebo-controlled phase 3 trial. And in order to discuss this trial in this journal club, our second panelist is Dr. Mrinal Patnaik, who is a distinguished hematologist, expert in MDS and myeloid malignancies at Mayo Clinic, Rochester. So you can see here the summary, the abstract, which was published with this paper. It's an international trial, 17 countries in adults. And these were cases, and we will discuss the selection criteria with low risk MDS who were randomized electronically to receive the experimental drug, namely Imetelstat or Imetelstat, or a placebo. And actually, the primary endpoint was about the 8-week red blood cell transfusion independence. And we will discuss, obviously, the strengths or weaknesses of such endpoint. And actually, the authors should be really commended for doing this because the trial was run in the middle of the COVID pandemic between September 2019 and October 2021. And they have included and treated 178 patients, 118 in the experimental arm, 60 in the placebo. And to make a long story short, it looks like that this drug offers a novel mechanism of action with durable transfusion independence compared to placebo. And you can see here, this is one of the figures of the trial showing the 8 weeks red blood cell transfusion independence. You can also appreciate the durability at 16 weeks, 24 weeks, and beyond 1 year. Also, you can see here on these curves the percentage of patients over time between the two arms with over than 8-week red blood cell transfusion independence. When it comes to safety profile, actually, I would say, but of course, we will discuss this with our panelists, that the safety profile was rather very good, in my humble opinion, and these are more manageable, I would say, side effect. And the editors of the journal actually concluded that this trial represent clinically meaningful. Actually, this new drug would represent a clinically meaningful and statistically significant improvement in red blood cell transfusion independence in this population, and probably it will represent a key player in this field. So I will stop my summary here, and I will now direct, as usual, the first question to our guest, Dr. Mrinal Patnaik. Thank you for joining us, and good morning. I know it's 11 a.m. for you in Rochester. It should be cold by now. I guess snow should be somewhere. And before going into the details of the protocol, Mrinal, I would kindly ask you to remind us what are the current challenges, therapeutic challenges we face when it comes to the management of low-risk MDS, and then we'll discuss the paper.
Thank you, Dr. Mohty, and good afternoon or good evening, everyone. It's a pleasure to be here, especially with Amer, a close friend and colleague. Like to underscore that this paper actually was accepted in Lancet and not Lancet Hematology or Lancet Oncology, just highlighting how important the
Apologies,
study. Yeah, no, no,
if
yeah.
I said something wrong, I do
No, no, no.
apologize.
I just want to tell you how pivotal this study is. And I was fortunate enough to, write the comment and review the data. I've also used this drug back in 2013. Mayo Clinic had led the myelofibrosis study, and we had done a pilot in RARS, RARST. So I'm quite familiar, with the whole development process. And I'm really proud of Amer to lead a trial, as you said, during the pandemic to lead us where we are. The word low risk MDS is a misnomer. So while these patients don't necessarily have the mortality with high risk MDS, the morbidity and the impact on quality of life is significant. So these are older patients, usually they have pervasive inflammation related to the MDS and transfusion dependence, whether it's red blood cell, but in sometimes platelets or both, significantly impairs their quality of life. Now, fortunately, we've had teams like Dr. Zeidan's team dedicatedly working on trying to find solutions for this patient population, and we have made progress. Dr. Zeidan has also been part of the team that led to the approval of Luspatercept. As you're aware now, with the COMMAND study going beyond just ring sideroblasts, patients with MDS with deletion 5q have options with lenalidomide, but a vast majority, are subjected to either short durations of responses to erythropoiesis stimulating agents, or many of them are actually ineligible because their baseline endogenous EPO level is so high, given the anemia, the inflammation, the hepcidin deregulation that exists at baseline. So while we have options, there's still a large number of patients who continue to remain anemic, continue to remain transfusion dependent, or who develop these issues after responses to the agents that I just described. So this sets the stage of understanding why we need drugs for these patients. Chronic anemia has huge impact on quality of life and on physiological outcomes. It leads to resting tachycardia, cardiac dysfunction, demand ischemia, cognitive decline, and just overall functional decline because of that. And then whether or not they get transfusions because of the altered iron balance over time, there's significant iron overload as well as free radical damage to several organ systems. And since these patients have a longer survival compared to those with high risk disease, iron toxicity becomes a serious issue that a lot of them have to deal with. And although iron chelation is an option, it's not always well tolerated. And sometimes it cannot just catch up with the amount of transfusions these patients need. So I think this really sets the stage for the need for medications in this realm, which is why I really feel that imetelstat is an important addition to our armament, and we hope that this can get regulatory approval soon.
Excellent. Thank you so much. That was very clear. Now, Amer, congratulations for this really fantastic paper, which I think is going to make a difference. But before going into the detail, can you please remind our audience what is this new drug, Imetelstat, the mechanism of action and the design of your trial?
Yeah, this is a great question. I actually want to start first by letting that audience know that there was a really fantastic commentary written on the paper by Mrinal, and I think it covers very important aspects related to the journey of this drug as well. Beyond this, hopefully being approved in next year as a novel in class. This is first in class. This is the first time we have a telomerase inhibitor potentially being put to clinical use, but this drug actually had a turbulent pathway, which is not uncommon in our MDS drugs, unfortunately. And it was studied initially with ET and myelofibrosis as well as MDS. And at one point, actually, during the development, the main collaborator on the drug, which was Janssen, pulled out of the agreement with the smaller biotech Geron. So Geron had to continue the drug development. So that drug could have been derailed very easily from a developmental point of view, and then it hit Corona. So I'm particularly actually proud of the entire effort by the sponsor as well as the investigators and the patients to take this all the way to a very successful phase 3 trial, in my opinion, and I anticipate approval by mid-2024 when the drug completes its review by the FDA. So the drug is, as I mentioned, is a first-in-class telomerase inhibitor. So many of your audience are familiar with telomeres. So telomeres basically are nucleotides arrays that sit at the end of chromosomes, and what they do is they prevent chromosomal breakage during proliferation. So they are very important for proliferative cells. And one of the mechanisms by which cancer cells proliferate and avoid dying is by increasing or maintaining the length of their chromosomes is by increasing the expression of telomerase, which is the enzyme that maintains the telomeres and therefore prevents the chromosome from breakage, which will lead to cell death. So the main idea of telomerase initially was focused on the idea of synthetic lethality, that you are exploiting a therapeutic window where rapidly proliferating cells, which are dependent on having overexpression of telomerase, which was, by the way, shown in also in patients with MDS, and that would this leads to selective killing of the clonal malignant cells while allowing the normal or residual hematopoiesis to re-emerge. So this was the theory. The drug is given intravenously because it's, as I mentioned, it's an oligonucleotide. It basically binds to the RNA template of the telomerase, so it cannot be taken orally. It has to be given IV infusion, as you mentioned during the introduction, over 2 hours. Is given every 4 weeks. And the dose initially actually was at a higher dose at 9 milligram. This was in the MPN experience, but there were some concerns about liver issues and count suppression. So based on that, when the drug moved to the MDS world, which happened again early on, as Mrinal mentioned, they had one of the first experiences in Mayo Clinic that actually showed that there is a tendency for patients who have ring sideroblast to respond, which was confirmed in this phase 3 trial that was just published. So the drug is given intravenously every 4 weeks. The Amer study that we published actually had 2 parts to it. So the first part was a single-arm phase 2 study that was published 2 years ago, I believe 2020, led by Dr. David Steensma, in which they showed that the drug was active in a pretty high number of patients, 40% of patients achieving transfusion independence. But those patients were not exposed to hypomethylating agents or lenalidomide. So those are patients who only have received ESAs. And based on this, the phase 3 part of the study, the Emerge, was conducted, which was, as you mentioned, a randomized placebo-controlled double-blind study. So we did not know, the patients did not know whether they were getting the drug or placebo. Importantly, the eligibility was similar to uh, uh, to the phase 2 in terms of focusing on patients who were not receiving hypomethylating agents and lenalidomide. The reason why I emphasize this is because we technically don't know the activity in that setting, and this is one thing that we should study. And another important thing to emphasize on the eligibility, that those patients were heavily transfusion dependent. So most MDS trials will accrue patients who receive 2 units per 8 weeks. This trial actually required the patient to receive 4 units or more per 8 weeks, and the median transfusion received on the trial was 6 units per week. That's almost 1 unit of blood every single week. That's a lot of blood. I can tell you those patients tend not to respond really to anything, let alone achieve transfusion independence. So when the end result came with a transfusion independence rate of almost 40%, which again is unusual because it was the same what was seen in the phase 2. Usually once you go to phase 3, almost always the response rate would come down lower. So to me, I wasn't surprised by the result of the phase 3. I was expecting the drug to be active, but what was a bit surprising to me is that the efficacy maintained from the phase 2, which is not something that you often see, but also because the median transfusion baseline was very heavy. So to achieve transfusion independence was, I think, quite impressive. And we can talk about the side effect profile, if you want.
We will come to this. Thank you, Amer. Really very state of the art, I would say, design. And so here, of course, by the way, I would like to remind our distinguished audience and participant that, of course, you can post your questions, and we have already a few of them. I'll handle them in a few minutes. Don't worry, guys. So, Mrinal, now I would like to, it looks like you're positive, but maybe I'd like to get your opinion and a clear analysis of what you think about the efficacy and safety of the drug based on these results.
Yeah, no, thank you. As I said, I think it's clearly an area of unmet need. So, this is something that's going to make a huge step forward. Also, as Amer pointed out, this is the first in class of these Mer oligonucleotides coming into, a human disease entity that may achieve approval and would definitely trigger, further research and optimization of these molecules. So we have to keep in mind, that this is just going to open the door into this field. As Amer pointed out, the area that is of great interest to me is how does this drug actually work? So if you look at telomeres, as Amer pointed out, these are hexanucleotide repeats at the end of linear chromosomes. And so take you back about 15 years ago when 3 individuals got the Nobel Prize, Carol Greeder, Elizabeth Blackburn, and Jason Swastak, for discovering telomeres in 2009, and they discovered it by looking at tetrahymena, which is the pond scum. So tetrahymena has 92 linear chromosomes, and it can completely cover the pond by rapidly dividing. And they were just astonished that the genomic material could maintain its integrity across so many mitotic divisions. And so these telomeres, they prevent premature shortening. And in the absence of telomeres, we see very fatal inherited disorders called as telomere biology disorders. The prototype is dyskeratosis congenita that pediatricians see, but we see a lot of adult patients with this. So the hypothesis is that, in MDS, because it's a neoplasia, the telomere lengths are shortened to start with. And this is not because of an inherited shortening, but it is because of replicative stress. And this leads to a compensatory increase in the telomerase activity and the expression of the holoenzyme. So TERT and TERK. TERT is the reverse transcriptase. TERK is the RNA template for the telomere, and this forms a holoenzyme. And so when you give an oligonucleotide, which is a template to the TERK, it kind of blocks the ability. And so when you aren't able to compensate, so normally telomeres vary between 5 to 15 kb in length. When they get below 2 kb, they reach what's called as the Hayflick's limit. And when you reach Hayflick's limit, the cell cannot divide. They go into senescence, and then there is SAS protein release and apoptosis. So if you look at the study here, they showed that independent of telomere length, TERT activity, there was a response. So this tells you that, this drug clearly outperformed the placebo, and regardless of any biomarker. But those that had increased activity were more likely to respond. So I think, what we would like to do here is obviously continue to better understand, better select patients. But as Amer pointed out, even without selecting or without biomarker, when you take these heavily transfusion dependent patients, to see not just the response, but the durability of response is encouraging. Now, when we come to the safety of the drug, I think, as you said, that there were significantly more grade 3 cytopenias compared to placebo, which is not a surprise. The drug is myelosuppressive. So unlike, luspatercept or darbepoetin, erythropoietin, which do not necessarily cause cytopenias, but more in line with hypomethylating agents or similar drugs. I think while we're treating anemia, the providers have to be in tune to be aware that these patients can develop neutropenia and thrombocytopenia, and so how to manage that, how to watch for that.
So we'll come to this, but because you already mentioned the biomarker issue and who can respond and maybe who cannot respond, we have a question here, Amer, from the audience about whether there are some specific subgroup with some chromosomal aberrations or molecular changes that could be expected to respond more or less to this agent. Any thoughts on this? I don't believe it's the case, but I prefer to ask you.
No, you are correct. So the study did a subgroup analysis looking at the things we typically look at within the context of MDS, which is or lower risk MDS, which is the transfusion burden, and the drug worked whether you are lower or higher transfusion burden. The presence of ring sideroblast. Patients who have ring sideroblast seem to respond better to luspatercept, to imetelstat, to whatever it is, they seem to respond better. So we saw the same thing here. This other thing we saw is the APO level, and also those patients responded regardless. One thing I would say, and we did not mention this as opposed to ESA and potentially luspatercept, is that this drug potentially has disease modification impact. What does that mean? So in lower risk MDS, the traditional endpoint, as we discussed, is transfusion independence, right? It's more of a palliative or symptomatic kind of relief of transfusions. But ultimately, what we want to achieve is to prolong patient survival, reduce the progression to acute myeloid leukemia, things that we have not historically been to achieve with any drug for lower risk MDS, partly because we did not have good drugs, but also because the trials were not designed statistically to look at these things. So with this particular drug, we are seeing hints of disease modification. For example, we are seeing lower number of ring sideroblast with treatment. So it's not, again, improving the transfusion, it's lowering the ring sideroblast. It's leading to more cytogenetic responses, meaning that there is clonal clearance, there's reduction of the variable allele frequency of some of the common mutation that is also associated with transfusion independence, suggesting that this is the mechanism by which the drug works. So why is that exciting? Because it potentially opens the door that this drug might not only improve the hemoglobin and reduce the transfusion need, but also it might lead to reduction in progression to leukemia or prolonging overall survival. Of course, these are things that are going to require longer follow-up. I think the other point to kind of highlight that this drug, the improvement in the hemoglobin was very, very high. So most of the lower risk MDS studies, the primary endpoint would be that the doctor stops transfusion. So when you look at the hemoglobin difference, for example, with ESA or even with luspatercept, the hemoglobin increases around 1 gram. So those patients go from 8 to around 9. With this drug, with imetelstat, the patients went all the way from a pre-transfusion hemoglobin of 8 for patients who responded all the way to 11. So it's a very huge increase in the mean hemoglobin rise, and it was sustained for almost a year. Again, all of these indicate that the drug is probably not hormonal, I guess, in terms of how it works. It's probably doing something at the level of the disease. But I also agree with Mrinal that while we think of it as a telomerase inhibitor, there could be other mechanisms by which it acts. There has been other presentations actually in EHA looking at the immune environment modification. So I think this needs to be fully explored. This has not been an infrequent theme in MDS. Lenalidomide was the same way. It was introduced in MDS, and every few years we found a new mechanism by which it would work. So I would not be surprised if with this drug, um, we are going to find new mechanisms. And as my Dr. Kantarjan, I always like to quote him on this. He always says, like, the real research starts once the drug is approved. So I think hopefully once this drug is approved, and this is going to open the door for second in class and longer follow-up, bigger studies, etc. I think this is going to really allow us to understand the biology of how it works in a better way, and hopefully allow us to use it in a more directed way. But the short answer, this is a long answer to say that there isn't a specific subgroup of patients in which the drug does not seem to work.
Excellent, and it looks like the drug has some exciting perspective. However, Mrinal, you mentioned, and I believe this is a weakness, the issue of grade 3 and 4 cytopenia. And we have, for instance, a comment from a colleague who has patient included in this study, and he mentioned that injections needed to be delayed because of this kind of side effects. What are your thoughts, and is there any way to circumvent this? And we have, for instance, maybe use of GCSF or something else?
No, this is a great question. And, I've, as Amer pointed out, the study that we did at Mayo Clinic was at a higher dose. It was 9.4 compared to the 7.5 that was done on iMerge. And in that study, we saw significant issues, with prolonged cytopenias, significant delays in treatment. There were some patients that, in particular, would have significant cytopenias with cycle 1. The delay would be, for a few weeks, but then once you got them back, either at the same dose or a lower dose, they would tolerate it better. So I feel that, while there was no mortality in the iMerge study related to this, it is something that we would need to watch for carefully. We may have certain subsets of patients that are more susceptible to cytopenias, is what I have written in that commentary, that I don't think one size fits all for everyone. In particular, I'm concerned about patients who may have, not an overt manifestation, but may have short telomeres, either because of a hereditary shortening, or may have inherited short telomeres from a family which has TBDs. These are much more prevalent than we anticipate. Are they more likely to have prolonged myelosuppression? We don't know, but I think there are subsets that we would want to watch for. If somebody's marrow is fibrotic, if they already have a hypocellular variant because of immune mediated or germline issues like DDX41, will they do well? In terms of tolerability, we haven't studied that. So I feel that there may be subsets that don't do well, but overall, with the reduced dose that Amer highlighted, and the duration was also increased from every 21 days to 4 weeks for the MDS population. This may be better tolerated, but I've also mentioned in my commentary that I think we have to watch out for thrombocytopenia and neutropenia and their management, and educate ourselves how to do better with that.
Excellent. Thank you very much. And maybe a quick question, Amer, and a quick answer about these subgroups, whether we have a question about whether hypoplastic MDS or MDS with fibrosis, whether they respond to the drug or not. Any experience?
This is a good question. I don't believe we have looked specifically at those specific subtypes. The I think one caution in terms of when you think about the drug mechanistically, and this is more of a theoretical concern rather than something that we actually saw is that, as Mrinal mentioned, some of those patients evolve from a telomere disorder, right? Like you could have this keratosis congenita or some other telomere disorder that evolves into MDS. And theoretically, we think those patients could potentially, using the drug would not be a good option because they already have short telomeres, and that can cause, of course, other problems. But so I think we need to study this particular subgroup. But on all the other subgroups, I think as long as you meet the eligibility criteria, which I have to remind people, you have to have good neutrophil and good platelet level, because again, the drug can cause thrombocytopenia and neutropenia. I think with hypoplastic MDS with fibrosis, as long as they are lower risk and have good platelet and neutrophil count, I think it's reasonable to study them. We are going to look at these subgroup analysis and secondary analysis. And I also wanted to add to Mrinal kind of discussion about the side effects is that those cytopenias were generally reversible, like meaning when you hold the drug, they would the platelet and the neutrophil count would come back, and they have not led to an increased incidence of severe complications, bleeding, infection that are severe. However, that does not mean that you just dose the patient and you forget about them. So I think I tend to think of it more like trying to use a low dose hypomethylating agent, where you certainly can have neutropenia and thrombocytopenia, but you have to manage it by delaying the drug, reducing the dose, maybe prophylactic antibiotic, etc. So this is different than what you do with, for example, Luspatercept, which tends not to reduce the platelets and the neutrophils. Actually, in some occasion, it increases them. And I think in my mind that brings up a very important question: is how does imetelstat works after Luspatercept, because of the command trial that you mentioned or Mrinal mentioned at the beginning. The landscape of management of lower risk MDS is changing very quickly, and how do you sequence the drugs or potentially combine them is very important. We are borrowing some of your experience in the multiple myeloma, because this is what I think the field is heading into in terms of lower risk MDS therapy.
Excellent. Thank you very much. Now, that brings me to one key question, and we have several comments and questions about this. And this is, I would say, the elephant in the room, is how do these results compare to the results of Luspatercept? And what are the next steps in terms of combinations, for instance, with EPO, with Luspatercept, in order to further improve or optimize these results? Any thought on this, Mrinal?
Yeah, I think that's a great question. As Amer said, I think the first step is to understand whether imetelstat will maintain its efficacy and the response rates in patients who have been exposed to luspatercept. So I think that would be there was a subset, there were a few patients on iMerge that, they have reported on, but I think we need larger numbers because, when you modulate the TGF beta SMAT signaling, and you progress on that, are you still lending yourself to similar efficacy? So with the command study and the broader applicability, availability of luspatercept, I think that would be agenda item one. I think, with regards to synergy, we'll have to do preclinical. We also have to make sure that safety is not compromised, so in this particular group of patients, you have to remember that they were either EPO ineligible or refractory. So combining EPO with imetelstat would mean you're bringing this up frontline, or do we not discontinue the EPO if they've become refractory and continue imetelstat? That was done with lenalidomide on the SWOG study eventually, and actually Dr. Sakaris led the S1109 study showing that even once you become EPO refractory or ineligible, continuing with len gave you an advantage in terms of response without an increase in venous thromboembolism, which one would be hesitant about. So I feel that these have to be explored, whether we combine them with luspatercept, EPO, or use them sequentially, or as Amer said, with disease modifying effect, is there another combination that could be more useful in this regard?
Excellent. Thank you very much. We're almost at the end of this journal club. It's really fascinating because we're talking about a new family, something that will be, I think, practice changing. So now after this phase 3 trial, the approval of the drug, hopefully in the next few months, Amer, what are your perspective? What are the next steps? Because as you nicely said, and you quoted Dr. Kantarjian, approval is the first step, but then the rest of the work remains to be done. So what are the next steps? What are your plans?
Yeah, so for this particular agent, Imetelstat, I think, so there are a couple of other trials that are being done, like in higher risk MDS and AML after HMA failure, as well as actually a large phase 3 trial in myelofibrosis, which I believe is the only trial looking at OS as an endpoint. So this also could potentially be a major change if that trial is positive. But I'm anticipating a lot of those studies that use it as a combination partner, potentially to be done mostly after approval for logistical reasons. But for the entire field, I think we are moving in a direction, as I mentioned earlier, where we are using drugs, I think, in combination, trying to understand the sequencing of it, but also in earlier lines of therapy. For example, Command's trial already moved Luspatercept after ESA failure, which is the same what was studied here with Imetelstat. Command's moved Luspatercept as a frontline agent because it has beaten ESA. And while there's a question about the RS ring sideroblast negative patients, I think it's a very well established frontline option at this point. And now, for example, with Luspatercept, the next question that's being studied is, can you treat patients who are not transfusion dependent? So there's already actually an ongoing trial that just started called the Element trial, which randomizes patients who have lower risk MDS, who have symptomatic anemia, but they are not needing transfusions to receive Luspatercept versus placebo. And there's a whole category of conditions that Mrinal and I are also very interested in, called CCUS, which we think of them as precursors to MDS, although you could debate where does CCUS end and where does lower risk MDS start. But I think those conditions which don't have dysplasia but otherwise have like symptomatic cytopenias are potentially on the table to be studied for those drugs. So overall, I think lower risk MDS in 2023 has seen tremendous progress, and I only anticipate this to continue. There are a large number of phase 1 agents that are being also studied in this space, and approval of any drug always will bring, second in class and best in class and oral versions, etc.
Thank you very much, Amer, and I share really your enthusiasm. It's really a fascinating era. I do remember from my medical school 30 years ago, the low-risk MDS chapter was the easiest one because our professor used to say, well, all you need to know is that they need transfusion, and that's it. But now it's a true revolution, both when it comes to better understanding the pathophysiology, and this is driving innovation. And actually now we have more choices, and I believe having more choices to treat these patients, because we know how difficult and how the negative impact of anemia on the quality of life of these patients. So I'm very grateful to all of you guys working in this field and trying to advance things in the right directions. So thank you, Amer. Thank you, Mrinal. It was a true pleasure to have this journal club with you. Thanks to all our audience, and I hope you have enjoyed it as usual. It's the last journal club of the year 2023 because we will have the Christmas and end of the year break, but we will start the journal clubs again as usual twice per month, early January. And if you'd like to stay with us for today, I think in 20 minutes or so, we'll have the ICH is organizing the post-ASH roundtable debate dedicated to the latest advances in multiple myeloma from ASH 2023. But wherever you are, whoever you meet, wherever you go, please stay safe and keep well, and see you soon. Bye-bye.
Happy New Year. Bye-bye.